Okay, I'm ready.
Okay. Welcome everyone. It's a big pleasure for me introducing Dr. Loren Williams, in my opinion, one of the most important researchers in the order of life that you have nowadays. Loren worked with the ribosome evolution and many other things, important things to understand the origin of life. Loren, thank you very much for staying here with us. And I'm sure that will be a great conference.
All right. Thank you very much for inviting me. Um, shall I proceed then?
Yes, you can
Okay.
start.
All right. Okay. Yes, I want to talk to you today about our work about the ribosome and sort of explain to you why we think the ribosome is such an important uh, structure for us to use to understand evolution. We can understand many, many things about evolution with the ribosome, using the ribosome as our tool that we could not hope to understand in any other way. And I do want to acknowledge my research group here. I have many talented people. I'm going to talk about their work. Anton Petrov is here. He's a research scientist who's been with me for quite some time. This is Claudia Alvarez here. She's a NASA postdoctoral fellow in my lab. And this is Jessica Bowman, who is my lab manager and is a research scientist. But many other people have contributed to the things I'm going to talk to you about. So I want to talk to you about the ribosome. And if you look here, the ribosome is translating. And I'm going to be talking about the large subunit, which is where the peptide bond is made. I'm going to be talking to you about the small subunit. And the small subunit is where the decoding center, where mRNA is decoded. And I also want to talk to you about the nascent peptide chain and the exit tunnel. So those are sort of key things I'm going to be talking to you about in this talk that we're going to come back to. And just so you can see, the exit tunnel and the nascent chain are going to be an important part of this talk. So these are crystal structures of, this is a crystal structure of the ribosome, and this is the tRNA here. And down here at the bottom, that little red thing is a little piece of the mRNA that is interacting with the tRNA in the decoding center. So that's where the decoding is taking place. And then up here at the other end, we have, this is called the CCA of the tRNA, and this is the nascent polypeptide. This is a newly synthesized polypeptide that is coming out a tunnel. And the tunnel is very important when we think about the evolution of proteins and some things I'm going to be talking about. I'm going to be talking quite a bit about the tunnel. So the nascent polypeptide comes through a tunnel. And that's a really, really important part of the ribosome that I'm going to come back and talk to later. So the ribosome is very complicated. You know, this structure is 1,000,000 Daltons, very large. There's about 5,000 nucleotides of RNA we're looking here, and about 50 proteins. So it's a really large. This is the bacterial ribosome. The molecular weight is about 1,000,000. It's very complicated structure, very complicated function, all the things that go on. But one of the kind of amazing things I think about the ribosome is the chemistry that it does is very, very simple. So what's happening in the ribosome is you're starting here at the top where you have what's called the charged tRNA. That's on the left. That's an amino acid on the end of the tRNA. And then you have this is the nascent polypeptide coming here. And really, like sometimes people will say the ribosome makes peptide bonds. Okay, that's not really correct. What the ribosome does is it does a transfer reaction. It transfers the nascent polypeptide from a tRNA up here. And so this is called ester amide exchange because we have an ester bond here that is being converted to an amide. So it's ester amide exchange. And this is the simplest reaction. It's actually hard to make a peptide bond, but it's very, very easy to convert an ester. In fact, if you take hydroxy acids and amino acids and you dry them down, just evaporate them out on the bench at room temperature, you can do ester amide exchange and make peptide bonds in that way. So this is the chemistry of the ribosome. So simple, and this actually speaks to the age of the ribosome. This core functionality of the ribosome is old. It's older than biology. And this is the chemistry from before biology that we're seeing. It's not a sophisticated enzyme at its catalytic core. So, okay, so why do we care about the ribosome and why do we care about translation? Okay, first reason is, like I said, the ribosome is very old. It's as old as any mineral. So, you know, the entire surface of the Earth has been remodeled while the ribosome has been sitting there unchanged. The ribosome is about 4 billion years old. It's very difficult to find any minerals that old. It's the oldest system in biology. So if we want to look back deep in evolution and try to understand some of the earliest and most profound effects in evolution, the ribosome and the translation system are how we do it. It's also essentially the only part of biology that's truly universal. If you want to talk about things that happen in you and in bacteria and archaea and yeast, the only thing that is happening the same in all of those is translation. So it's the universal part of biology. And when you talk about economy, you know, translation, it really depends. Even with, let's say with cattle, when you want to know, how good is my cow at making milk? Really, you define that by translation, by the synthesis of proteins. Or when you say, what for an E. coli to actually replicate itself, what does it have to do? It has to make all of its proteins. That's where the vast bulk of consumption goes into this production of proteins. And so the translation is really almost the definition of biological economy. And translation is also extremely complex. I mean, I showed you that chemical reaction. You know, the ester amide exchange is a simple reaction, but everything building up to that, you know, the decoding, the synthetases where you put the tRNAs on the amino acid. There's so many proteins, so many components. If you think of everything involved in translation, it's really I think it's biology's greatest achievement. And what I mean by that, if you just think of DNA replication, you know, you're taking one polynucleotide and you're using it as a template to make another polynucleotide, and you use Watson and Crick base pairing. I mean, it's complicated, but it's not that complicated. You can understand it. But when you think about converting the information in a polynucleotide to a polypeptide, it's a completely different chemical space. There's no direct pairing. There's no direct molecular interactions. It's all very subtle and indirect and just outrageously complicated. So translation is just inherently far more complicated than other parts of, you could say, the central dogma. You know, if you compare it to transcription or replication, translation is just like far more complicated. It is the hardest thing I think biology has ever done, is figure out how to take one polymer of a defined sequence and translate that into a different kind of polymer that is chemically unrelated. This is biology's greatest achievement, I think. And also, we think about centrality. When I think about sort of how I think about biology, to me, biology is, and Savio has written about this also, I think he might agree with me, is biology is it's a consortia of molecules in mutualism relationships. You know, protein takes care of RNA, membranes protect things, all these components, it's a mutualism relationship on the level of molecules. And the ribosome is sort of the center of this, you know, where RNA is binding to protein. The ribosome itself is a cooperative arrangement of RNA and protein, and it synthesizes protein, and then that protein forms polymerases and synthesizes RNA. So this is just a big cooperative system of molecules sort of at the center of the biological universe. So when we think about evolution and the tree of life, I mean, we don't have to always think about universal evolution. Sometimes we're just thinking about fish or mammals, but if we want to think about an evolutionary scheme that involves everything, there's only one way to do it, and that is to use the translation system. And like I said, the translation system is the only system in biology that's truly universal. So if you want to make a tree of life that has everything on it, you don't really have any choice. You have to use the translation system. And this is why Carl Woese, who discovered, the third branch of the tree of life, the archaeal kingdom, it's why I think, one of the reasons why I think he was so smart. I mean, he somehow figured out ahead of time that the translation system was the way to understand these deep questions in biology. And we've all been following Carl Woese in one sense ever since he did that. He had that great insight. So the questions we're asking when we think about the ribosome, number one, like, where did the ribosome come from? I mean, it's huge. There's so many players. It's so complicated. Seems inconceivable, but it had to evolve. It had to come from something. And we spent a lot of time thinking about where did the ribosome, there's sort of two limiting models. One is called the RNA world. In the RNA world, you had like all RNA. And then that RNA kind of invented the ribosome because proteins were better enzymes than RNA. An alternative model, which I kind of favor, but we don't really know this, is sort of a coevolution model where polypeptides and polynucleotides just co-evolved from the ground up together, and that the ribosome was really one of the first things biology did. So those are deep questions that we're trying to address in our research, like where did the ribosome come from? And of course, we're thinking about the origins of the genetic code. Actually, to tell you the truth, I don't really think about the origins of the genetic code very much. And people ask me about it a lot, but my feeling is that once we understand the origin of the ribosome and the evolution of the translation system, then we will understand the origins of the genetic code, but it will never happen in reverse. We have to really understand the origins of the translation system before, as a whole. We can't just focus on the genetic code. So and we're not ready to do that. So I actually don't really think about the origins of the genetic code too much. One of the things we think about a lot is asking questions, how did proteins learn to fold? How did folding initiate, and what are the mechanisms of fold evolution? How do protein folds change? You have structures of proteins, and one will change into another, and how does that happen? One thing we know is that this was not done by brute force, in the sense of if you synthesize full chain proteins, full length proteins, and just start synthesizing and waiting to find one that will fold, you'll run out of carbon in the universe before you find one. So this had to be some directed pathway that did not involve this random blind search. And all these questions are related, but another really important question to us is like, what evolutionary forces are constraining the ribosome? Like the ribosome, I'm going to show you this later. I mean, it's been amazingly well preserved over 4 billion years. And really almost nothing else has. I mean, why is the ribosome so highly conserved, or the translation system? So there must be a lot of constraints on the ribosome, or we say a lot of dependencies on the ribosome that we don't fully understand. Because if you look at it and watch it work, you could say, yeah, I could change this or change that. It shouldn't matter very much. And yet biology over billions of years hasn't done that. So there must be something holding it back. Evolution will do anything if you give it a chance, just randomly, and there's some force being applied to the ribosome which just doesn't allow these things to happen. And so for example, we can compare the ribosome to RNase P. RNase P is also a very, very ancient RNA protein assembly. It's a ribozyme. and it's probably almost as old as the ribosome, maybe as old as the ribosome. And we're studying the sort of evolutionary trajectory of RNase-P, and we can see there's like some fundamental differences between the constraints on RNase-P and the constraints on the ribosome. So we're trying to understand these things. These are questions we're trying to figure out. And then this is a really interesting question. If you look over recent biology, the mammalia, even though the core of the ribosome is highly conserved, what I'm going to show you is that the ribosome is also undergoing extremely rapid change in mammalian systems. It's just exploding. It's like you stepped on a tube of toothpaste and it's just going out. That's what the ribosome is doing in a very rapid time scale. And by rapid, I'm talking about on the time scale of the ribosome. So rapid is over hundreds of millions of years. So it's on a relative scale, but it's still on the scale of the ribosome. Right now, it's undergoing more rapid change than at any time in the history of biology. And we're very interested in understanding what that's about. Okay, so we have the tree of life that's on top here, and there's three branches: a bacterial, an archaeal, and a eukaryotic branch. Or now we're starting to think that eukarya are just a sort of a little branch off of archaea. But we still say there's these three branches of the tree of life, and they come from this thing we call LUCA, which is the last universal common ancestor. And we don't really fully understand LUCA, but what we do know is that the ribosome and the translation system was essentially mature at LUCA. That's one of the few things we can say for sure about LUCA, is it had a well-developed translation system, which was very similar to an E. coli, reasonably similar to an E. coli. So that's LUCA. And then what we can do, and I'll show you this a little bit, is we can walk the ribosome back. We can see, people don't usually think about evolution in such a deep way. You say, what happened before LUCA? People, basically, they say, evolution started at LUCA, but it didn't, obviously. I mean, LUCA was a microbe of some kind. It was reasonably sophisticated. It had a fully functional translation system, meaning it had energy transduction. It had to have metabolism, probably compartments. So LUCA was a pretty sophisticated organism, or probably ensemble of organisms. And the nice thing about the ribosome is that it has allowed us to walk back deep, deep beyond LUCA and see things that have happened. So I'm going to show you a little bit about how we do that. Okay, so let's start here. How conserved? I told you the ribosome was conserved over all of biology. What exactly do I mean? I'm going to show you what this means. So this is a backbone trace of the ribosome from E. coli. And I've taken out we're not looking at sequence here. So when I talk about conservation, when people think about conservation, you can think about it different ways. A lot of times people think about sequence conservation. That's sort of a standard way to think about evolution. You can do multiple sequence alignments and calculate distance trees. But a lot of times we are thinking about structural conservation. And the reason why we think of that, we focus on this, is that structure is more conserved than sequence, or structure changes more slowly over evolution than sequence. Or to say it another way is, you can have divergent sequences while the structure is conserved. And in fact, if you think of a helix here, like there's these helical regions. Like here's a helical region. That helix is, as long as you have base pairing, the helix doesn't really depend on the sequence. You can have a lot of different sequences that will make that helix. And so when we talk about structural conservation, we're not limited to sequence conservation. So I'm going to focus on structural conservation for a minute. So this is a part of the bacterial ribosome. Now I've superimposed that on a backbone trace of an archaeal ribosome. So, this is what we're looking at here, is actually 8 billion years of evolution. Because let's say LUCA was 4 billion years ago. We have 4 billion years to the extant bacteria and 4 billion years to the extant archaea. So the total divergence is 8 billion years. And this shows you how much change you get over 8 billion years. Not very much. In fact, essentially none. And then if I put, just for comparison, I'll put the eukaryotic ribosome on top of that. And you can see really that it's gone nowhere. What I'm saying here is that every ribosome in every living system is going to look like this. And we're looking at the RNA here. So if we, I have another one here, but you get the point. Okay, so let's just look at, that's a ribosomal RNA. Let's look at ribosomal proteins. You know, the bacterial ribosome has about 50 proteins. This is a bacterial, this is ribosomal protein L4, which we like to look at. And this is from an E. coli, from a bacterium. If we look on the other branch of the tree of life, this is from an archaean. And I mean, there's two things I guess I'll say here. One is that it's not as conserved as the RNA. The RNA is, the ribosomal proteins seem to be a little more divergent over evolution than ribosomal RNA. But it's still, if you think about, you know, two proteins that have diverged, you know, at Luca, it's still incredibly similar. And if I then put the same, I'll put a yeast and I'll put a eukaryotic ribosomal protein. So this is basically shows you what L4 looks like in everything alive. I mean, this is the total divergence in all of biology is being represented here. And you can see there's a little shift. The alpha helices move a little bit and some things like this, but the extent of conservation is very, very high. Okay, so that's how conserved the ribosome is. But now let's talk about how variable is the ribosome. Like I told you that human ribosome, or the mammalian ribosome, is undergoing explosive growth, and there's a lot of variation in the ribosome. So let's, for a minute, talk about that. So if you look here, I have an E. coli ribosome, I have an archaean ribosome, and I have a eukaryotic ribosome. Same thing. I'm just doing the same trick over and over again. And the colored parts, like the red and the blue, those are the part that are universal, that are basically identical in everything alive. And then the differences are the black. And you can see like the differences between E. coli and Pyrococcus, between a bacterium and archaean, are pretty subtle. There's some black, but it's mostly blue or red. Then when you look down at the yeast down at the bottom, the eukaryote, you can see there's a lot more black. And what has happened is things are getting added on. So the Encerramento ribosome has RNA that has been added on to this, what we call the universal core. And so here I just kind of have blown that up and highlighted it. In fact, here I've actually added another one. So here we have E. coli, which is a bacteria at the top left. We have Pyrococcus, which is an archaean at the top right. Then we have yeast. And then I put our ribosome, your ribosome, which is the Homo sapien ribosome. And there, look how much black there is on that. So even though the blue parts are common, and even between yeast and human, the blue parts are essentially identical in 3 dimensions, once you look at those black parts, you start to see a lot of divergence. So that's the ribosomal RNA. What about ribosomal protein? Now I did something which was not exactly straight before. I showed you this protein, this ribosomal protein L4. This is from a bacterium. Then I showed you an archaean version, and I showed you the yeast version, but I cut something off. So now I put it back on. So what you can see here, it's the same as with the ribosomal RNA. When you get to eukaryote, things start to branch out. So the core structure is the same, but now I get these things that are added on. This is C-terminal, I think the C-terminal alpha helices are very, very common in eukaryotic ribosomal proteins like this. So, and if you look at a bunch of eukaryotic ribosomes, you'll see a lot of this stuff happening, and it's actually fairly divergent. So you have this common core, which is basically the microbial ribosome, and everything alive has that, and that's true for RNA and protein. But then you get these things added on, especially in eukaryotes. And in fact, not just especially in eukaryotes, but especially in warm-blooded eukaryotes. So this is sort of a phylogram showing ribosomal proteins. I mean, I showed one protein and I showed how things get added on, but there's another layer, which is that you have what are called the universal proteins, and those are in the black here at the bottom. So these are universal, these are the proteins that are universal to everything alive. And the one I showed you was called UL4. That's the one we looked at. That's a universal protein. But there are bacterial specific proteins. There are archaeal specific proteins, and all of the archaeal specific proteins are found in eukarya. Every one of these proteins here is seen there, and then eukarya has even more. So this is the sort of general pattern we see, that eukarya have bigger proteins and they have more. And of course, they have more RNA also. So I talked about the explosive growth of the ribosome. I just want to show you what that looks like. So this is at the bottom. Let's just look at the bottom. This is like size of the ribosomal RNA. We're ignoring the proteins for right now, versus time. And this is over 4 billion years. So we're starting here. And this is not very firm. I mean, I don't really know when life originated. You know, this might have been 3.8 billion years ago, or I don't know, maybe it was 4.2. I really don't know that. And I really don't know. All I know is that at some point there was no ribosome, and then there was a ribosome. And we don't know too much about what happened here. So that part is the most speculative part of that graph. But we think whatever happened, probably happened pretty fast. Anyway, and then there was this time we call the boring billions, where nothing happened for billions of years. This is when life was microbial. So the microbial world was just sort of happy with its ribosome and didn't do anything to it for billions of years. Then starting around in here, you had the rise of eukarya. The earth oxygenated, the rise of eukarya, biology started to do some different things. And then look what's happening here. And so let me just tell you, how do we know this? It's pretty easy to do reconstructions of ancestral ribosomes. I mean, you could do it with any system now. When you have a phylogenetic database and you can make trees, you could do ancestral reconstructions. And it's very sort of easy, and it's pretty firm. I mean, the sizes of these things are not really in doubt. We do have a very, very accurate, we believe, trajectory for the size of the ribosome. And what you can see here, I mean, look what's happening over recent biology. And recent biology, really, this is a couple 100 million years here, right? So we're over the last couple 100 million years. And this is, I need to say this also, I'm not sure I said this, this is the human ribosome. We're following the lineage, this is the history of the ribosome in humans, right? If I followed the history of the ribosome in E. coli, it would just be flat here. It's going nowhere, right? So, I mean, this is for our particular ribosome in Homo sapiens. So what we can say, maybe, is that if we wait 500 million years, our descendants are going to have a truly enormous ribosome. I mean, I don't know what's going to happen. I mean, if you project this out, it's obviously going to hit a wall somewhere, but it looks like the ribosome is going crazy. And so why is this happening? What is different in biology now? And what is different in mammals? Actually, we see this in birds and mammals. What is happening here? We're not really sure, but let me just show you a few things that might help us try to understand why the ribosome is exploding in birds and mammals. So I'm going to focus, this thing I have circled here is called expansion segment 7. And this is I think it's about 800 nucleotides in human. That's ours. I've got it circled there at the bottom left. And if you look at E. coli, it's just a little stem loop. And in archaea, it's a slightly longer bent stem loop, but it's really not very significant. But then this thing goes crazy in eukaryotes, especially in birds and mammals. So if you look here, this is the phylogenetic representation of eukaryotes, only eukaryotes. So on the right-hand side, we have fungi. These are all fungi. Over here, we have plants. I think rice. This is rice. Here's, some little bushy trees, some things like that. And then here we have animals starting here, going in this direction. And warm blood, here's a frog. This is where warm blood starts to kick in over here. And you can see, starting with the warm-blooded species, we start to get these things we call tentacles. See, this is a chicken. See that enormously long helical region. Humans, our tentacles are not as long as chicken, but we have more of them. So not only is the RNA getting bigger, but it's changing its character. These tentacles, I mean, really the human ribosome looks kind of like an octopus. It has these long tentacles of RNA sticking out. So this is a new phase of the ribosome, which has just happened over the last, started maybe a 250 million years ago, that the ribosome started to develop tentacles. So why did it do this? What is happening here? We have some ideas. In fact, let me just show you. We don't know the answer yet, but I think we're onto something here. If you look at, let's say, ES7, the size of that ES7 versus number of neurons, that's what I graphed here. You can see that there is a nice relationship. Now, you can see relationships like this. It doesn't mean anything is causing anything, but what neurons require is sophisticated transport. And, because neurons are so long, and you have to transport ribosomes in neurons. And we think that it's possible that that these tentacles are involved in transport. So actually, I have a student studying ribosomal transport in various systems, and we're trying to work out the function of these tentacles. So that's one possibility. It seems tantalizing when you look at this graph, but the graph doesn't really tell you the whole story. So that's just something we're working on. Okay, I want to look at ES7 for a minute. I told you that because not only does it grow over, like, let's say in the human lineage, it just grows and grows and grows, but it also provides us with a nice model system, an experimental system that we can use to understand how RNA grows. Because we have all these structures of different sized RNAs, and we can look at them and compare them and try to understand over evolution, how does ribosomal RNA change? And so I just want to show you how that is. And the reason I'm going to show you is because it turns out to be very, very simple. And I'll just show you this. So this is the smallest piece of RNA at that size. I showed it to you in a previous slide. This is the base of expansion segment 7. And then then this is in an E. coli. And this is our model for the LUCA. This is what we think it was in LUCA. Then in the descendant of LUCA, one of the first descendants in an archaean, it looks like this. And what you can see here is that helix 25 is still there, and something has grown out of it. It got bigger. It didn't go away, okay? And it didn't really remodel. Something just grew out of it. So this is what we see in essentially all archaea. It looks like this. And then archaea is, of course, the ancestor of eukarya. So we have a pretty good model for what the deep ancestors of eukarya are like. So then, what is the next step in the evolutionary process? We have a yeast. And so look at what you see here is that helix 25 is still there, never goes away. And then some things have grown out of it, right? It got bigger. But actually the archaean, it bent a little bit, but the archaean ribosomal RNA is there in yeast, its ancestor, but the yeast, something has grown out. And then we know that yeast is a pretty good model system for the ancestor of animals. And so look at this fruit fly. You can see that the ancestor is there, and then the fruit fly has grown out all of these other RNA elements. And so this is a very primitive animal. And then when you look at a more sophisticated animal like human, you can see there's essentially the fruit fly ribosomal RNA is there, and then human has all this additional stuff. So this is very nice for us because what it means is we can really understand how RNA changes over time. And the analogy we use, which is, I think, a very apt analogy, it's a very good analogy, is a tree. Because a tree grows in the same way that the ribosome grows, which is that the tree does not remodel every year. It doesn't have to rebuild anything in order to grow a new branch. And and things add on to a tree, right? You have a branch, another branch grows out, branches grow out of that. So a tree grows in a way that is very similar to how the ribosome has grown over evolution. And I can take it even a little further, since we're all familiar with trees and how they grow and what they look like. I could give you an assignment. I could say, take little stickers and date in a relative way everything on this tree. I want to know the relative age of everything on this tree. And you could do it, right? Because you know how a tree grows. You know that the leaves are the most recent things, and the small branches are more recent than the big branches, and that the oldest part of the tree, is right in the center. If I drill in there, that's, into the center of the core of the tree, I can see really a record of the origins of this tree. And it turns out, so this is the idea. If you don't have to watch a tree grow now in order to look at a tree and understand how the tree has grown. And that's sort of where we are with the ribosome right now. So we know what the ribosome was like at the common core. It was pretty similar to a bacterial ribosome. But we also can say, where did that come from? I mean, we can work it out, just like you can look at this tree and understand how this tree grew. We can look at the ribosome and understand how the ribosome. I've probably looked at ribosomes more than I've looked at trees. And I live in Atlanta. There's a lot of trees in Atlanta, and I love looking at trees. But I think I spend more time looking at the ribosome than I look at trees, because we can, just like you can understand the history of a tree. Like if there was a drought 30 years ago, the tree has recorded that in its structure, and the ribosome has recorded its history in the same way. So I'm not going to explain all this to you because it's kind of tedious and it will take me a long time, and it's published. So I'll just say that we have in a lot of detail worked out how to walk back in the ribosome, and also what we see when we walk back in the ribosome. We've done all that, and there's some papers here if you're interested, or we could have a beer sometime and talk about it. But I'm not going to try to go over it now. So you can either trust me or not. We can work out in a great amount of detail what happened, the sort of evolutionary processes that by which the ribosome expanded from the beginning. And this is our model. So on the ribosome is on the left, and then other things going along with that. So let me just say this, let's say it like this. When we see the evolution of the ribosome, number one, we can look back like this. This is your mother, or this is your parents right here. Those are pieces of RNA before there was a functional ribosome. And we know what they are. We know what they look like in 3 dimensions, and we have made them in the lab, and we're studying them. And kind of a lot happened before the interface. This is something that's really important, is, the ribosome has a large subunit and a small subunit, and we can see that we know when that, in a relative scale, when that subunit was acquired. So both the large and the small subunit got pretty big before they ever met each other. So they were doing things that must have conferred advantage to some kind of very, very primitive evolutionary system before there was coded protein, because you can't have coding if the two subunits are not talking to each other. So that's how far back we can see. We can see very, very far back. And the other thing is, by looking at the ribosome, we can see the evolution of protein folding, because that is frozen in the ribosome. So all the really old proteins are what we call intrinsically disordered. Then at the next step, we have these isolated beta hairpins, and then we have very primitive beta domains, and then those elaborate. And we basically establish the age of things here, and then we say what was happening, and we can see these structures in the ribosome. So this is all structural analysis. We're not looking at sequences here. We're looking at three-dimensional structures. So everything I'm showing you here comes out of a three-dimensional structural analysis of the ribosome. Okay, so I'm going to try to finish up quickly. I just want to tell you sort of something that I talked about, the tunnel in the beginning. And I want to go back to that because I want to walk you through the evolution of the large subunit. We're pretty confident in our model of the evolution of the large subunit, and we're making all these RNAs and we're studying it experimentally. But I just want to show you what happens. So I told you the tunnel was very important, but there was not always a tunnel. So in your mother, this blue piece of RNA, there was no tunnel. There was just a sort of a bent stem loop. But one of the very earliest processes in the evolution of the ribosome, essentially the first thing that happened was making a small pore. And this is bringing these two blue parts together, the most ancient parts of the ribosome. So there's something about a pore that was very, very important to the ribosome. So there's no what you would call translation at this point. There is no small subunit. There's no GTP binding site. There's nothing. There's just the tunnel. And the actual peptidyltransferase center is somewhat assembled. But I told you that's not really an enzyme, right? The chemistry that the ribosome is doing, you don't need a ribosome to do it. You can just dry things down on the surface. So we don't think the chemistry was all that important. We think whatever was happening without the ribosome. And then the ribosome came around and said, I'm going to change things because I'm going to make a tunnel. And so the first step in the ribosome evolution was this, making that tunnel. The second step, the tunnel got bigger. The third step, it got bigger again. And one thing I want to say is at this point, the ribosome is associated with a lot of polypeptide that forms beta hairpins. There's no globular domains. There's no folded protein yet. Okay, so the ribosome is associated now with beta hairpins. The ribosome at this point is associated with what we call IDRs, which means non-canonical, that's intrinsically disordered regions with non-canonical peptides. So when you go from this to this, the proteins start to fold. They don't fold domains, they don't form globular structures, but they form beta hairpins. And those beta hairpins are totally wrapped in RNA. So RNA is somehow chaperoning the initial folding of polypeptide into structures. So that's the yellow part. Then we add the orange, the next layer, and the tunnel gets larger. And then the next stage, the tunnel gets larger. So I want to just so you understand, we were not thinking about the tunnel when we figured out our model of ribosomal evolution. We were not trying to prove a model about the importance of the tunnel. We hadn't even thought about the tunnel. But once we had the model, we realized the evolution of the large subunit is driven by this tunnel. It's the only thing that happens from beginning to end. There's a lot of things that happen. You acquire the subunit interface gets acquired, GTP binding center gets acquired, a bunch of things happen. But the only thing that goes on from beginning to end is the building of the tunnel. So we think this building of the tunnel is what was driving the evolution of the ribosome. So why does the ribosome need a tunnel? What does the tunnel do? And I have to say it took us longer than I thought to kind of really think intelligently about this. But so this is the tunnel I'm showing you here on the left. And you know, the tunnel is a funny thing. You know what the tunnel is? It's a nursery for alpha helices. You can form alpha helices in the tunnel. You cannot form beta structures. You can only form alpha. And so what we're pretty sure, it seems really likely, is that making of beta structures is trivial. We can see beta structures back here when the tunnel was very primitive, and there's no alpha helical protein associated with this part of the ribosome. It's only beta. So making beta structures is pretty easy. In fact, we know that. If you make random coil peptides and put them in solution, they will precipitate as beta structures. I mean, when you hard boil an egg, you're converting alpha to beta. In fact, amyloids, that's conversion of alpha to beta. Protein wants to be beta. Given a choice, a polypeptide will almost always form beta. The sequence modulates it a little bit, but there is this driving force for polypeptide to form beta. But getting alpha is tougher. And so we think the tunnel was a device ultimately that allowed polypeptide to make alpha helix. Without a tunnel, you don't have alpha helix. Everything, if polypeptide spools out of something, it just forms beta. But if you put it in this nice warm tunnel, then you can't form beta in the tunnel. There's no room. And the polypeptide forms alpha. And there's a lot of crystal structures of alpha helices in the peptide, in the exit tunnel of the peptide. So we know this is what is happening in the tunnel. So why do we need this? Let's let us do another question. This is what I like about the ribosome. It makes you think about things you normally would not think. Why does biology need alpha helices? I mean, why are beta? You can make very good enzymes out of beta structures. Why do you need alpha helices? I've never seen this. I mean, I teach biochemistry. I teach about alpha helices and beta sheets. It never occurred to me before. Was like, yeah, but why do we need them both? Why did biology go to all that work to build that tunnel? Why do we need alpha helices? And if you look at protein structure, you can see the answer. If you want a motor, try to make a motor with beta. Biology has never done it, right? I think this is a V1 ATPase or something. But if you look at motor proteins, if you look at levers and rotors and mechanical things that transduce force in biology, it's alpha helices. In fact, if you just look at allosterism, where that's sort of a more subtle kind of a motor almost, it's dominated by alpha helices. So really, beta sheets can make enzymes. You can make essentially any enzyme you want with a beta sheet, but when you want to have long-term communication and you want to transduce force, I don't think beta will do it. So biology was under pressure, and the response to that pressure was the building of the tunnel. So this is one of the, I think, greatest evolutionary stories of all. Is biology figured out how to, biology invented a peptide or a polymer that made beta. And then it said, no, that's not good enough. And somehow, I'm presuming there was a lot of other polymers there, and we're only looking at the winners. Obviously, that's how evolution works. So there was probably other polymers that could maybe assemble in sort of beta-like structures. But there was only one winner because there was only one special polymer that could do both these things. When you think about what it requires for a polymer to make a beautiful beta sheet and then a beautiful alpha helix and switch between those things, that's a really special polymer. And that's the winner. That won this sort of evolutionary contest that gave us the ribosome. So in the evolution of protein, this is what we can see, and I haven't really justified this fully, but the ribosome shows us that polypeptides started with intrinsic disorder. That means these non-canonical things, and we think that true ancestors of those weren't even peptides. Maybe they were esters, or what we call deoxypeptides, which is combinations of esters and peptides. That's the ancestor, we think, of polypeptide, and you can't assemble that, because if you look at the structure of an alpha helix and a beta sheet, you cannot do it with an ester. So then the first step in protein evolution was sort of the purification of that backbone chemically into a homochiral peptide. Even forget the sequence, because essentially anything can make beta. Any sequence can make a beta structure. That's pretty much. So that was the first step in protein evolution. And then there was a proliferation of beta domains. And we can see these in the deep part of the ribosome. And then those proliferated all over biology. We can see those. They're called OB domains and SH3 domains. My student Claudia is mapping out how these very primitive beta domains that were really created on the ribosome proliferated all over the ancient part of biology in transcription and replication and in all kinds of places. And then the ascent of alpha. And that required building of the tunnel and rigidification of the tunnel and all these fancy things that happened. And now we're in a period of dominance by alpha. Alpha, I think, is the dominant secondary structure in protein now. And it is, if you look at motors and things like this, it rules. You know, look at this protein I'm showing you here. There's some beta here. I'm not saying there's none, but you can see this is predominantly an alpha helical structure. So this is what biology really needed to do to get us where we are right now. Okay, so this is the story, part of the story of the ribosome. You can see it makes you think about things that, like I taught biochemistry for so long, and it never really occurred to me to ask, why do we need alpha helices and beta sheets? And the ribosome, I love studying it because it makes you ask questions that are kind of crazy like that. Okay, so I really need to thank my group, and I need to thank you guys for listening to me. And I'm very honored that I was given this opportunity to talk to you. And I can answer questions or whatever Savio would like. Thank you.
Thank you very much, Loren, for this amazing conference. We have some questions. Fabricio.
Hi, Loren. So
Hey.
thank you very much. It was a wonderful talk. A lot of progress since your last talk, I think 10 years ago in Paraíba. So it's very nice to see you again. And I have some questions about the origin of life, and there are some ideas about the origin of virus. Some giant virus before the Luca would be something previous, like another experiment. Do you think that this kind of entities or organisms, they would have something different that we see today? Like you said, only the winners we see, but because we have a coevolution of life, so we just see this kind of beta alpha helices, the coding system. What do you think you would expect? Do you think any kind of relic we could identify today somehow?
Yes. Okay. Let me answer that question. That's a great question. And I think 5 years ago, I would have said, no, I don't believe that. But I've totally changed my mind. So let me tell you sort of the way we're thinking about it. When we think about evolution, we think about replication, right? I mean, how do you think about evolution without replication? And then you say, well, a virus, how does it replicate? It has to take over things. But you know what? This is the way we need to think about it, is that polymers, I mean, you cannot have replication without polymers, right? I mean, if you don't have an informational polymer and you don't have an enzyme, you can't do replication, right? So the question is, where did that come from? And the way I'm thinking about it is we are thinking about evolution in a way that is too much dominated by what we see in biology right now. This, I think, is holding the origin of life back horribly, is that we are so blinded by biology. So if you think about the RNA world, people say RNA sprung up and started to replicate and be a genome at the same time. Where did RNA come from? I mean, it's a winner of an evolutionary process. And that is a thing that I think people are and if you think about viruses, and a virus is an assembly of polypeptide and polynucleotide, which I believe came out of non-replicative chemical evolution. So I'm not sure I'm answering your question, but I would like to say that this focus on replication and on catalysis and on informational polymers in thinking about the origin of life, I think is misguided. I think molecules can evolve. And the reason I'm so sure of this is this is what we're doing in my lab right now. We have figured out how to do chemical evolution without polymers and how to how to get to polymers. And this is an evolutionary process. You know, we need to think about evolution in a much broader way. This is great. This is an evolutionary talk, because I want to tell you, I think evolution transcends biology. I think chemists have failed to understand evolution, and that there can be evolution in chemistry. I know this because we're doing it in my lab right now, and we're writing up a paper on it. So, so viruses, you know, you don't need polymerases to do evolution, and viruses, you know, I'm not really explaining to you how I think this would work, but I think viruses could have come out of chemical evolution at the same time as other systems, or maybe even in advance.
Yeah,
Or
quite.
not viruses, the ancestors of viruses, of course. I'm not talking about viruses, but
yeah, yeah. We have still to figure out this. Maybe synthetic biology can get some clues, no? But we have a question here from a professor of biochemistry, Gloria Franco.
Okay,
so
great.
It's written here: when and how did the genetic code existing in the mRNA start to be identified as information to assemble amino acids into a informative polypeptide sequence?
Yes, okay. So this is sort of going back to what I was just saying, which is that when you think about how people think about the origin of life and evolution, you know, you say you have to have informational polymers. You can't have informational polymers without energy transduction, right? I mean, how do you can't, the ribosome doesn't work if you can't drive it, right? It's a motor. So the question is, where did all this stuff come from, right? It seems like everything is so interdependent. And if you start to say what happened first, you immediately just run into a wall, right? I mean, because you can't have metabolism without information. You can't have information without metabolism. Everything is dependent on everything else. And so, and the genetic code, you can't have information without a genetic code, because how are you going to replicate that information without enzymes? So everything ends up being tied up in a knot. And so what we started to think is that, you know, nothing came first. You know, one of the beautiful things about biology is how things can be linked together. And the analogy I use is an algae and a lichen and a fungus in a lichen, right? Those are inseparable biological systems that live in mutualism, and if you separate them, they die, and they create each other, right? So we think this is how molecules happen, so that RNA and protein and DNA, you should never think about which one came first. They all came together in a coevolutionary process that we don't understand. I'm not saying we understand it, but that there is a thing called chemical evolution in which molecules can evolve, and all of these things came together in a very sophisticated way. And so, and the genetic code, I would say, is the final part of that process, you know, but it was an incremental thing. Like we can see the large subunit was doing something before there was a genetic code, because it was doing something before it ever even saw the small subunit. And then we think the original function of the small subunit was really as a cofactor to make maybe slightly longer peptides, but not of specific sequence. And then that cofactor slowly evolved into a system of coding. But that it was the synthetic machinery, and the RNA was pretty mature before coding became very good. And coding didn't have to be very good maybe at the beginning. You know, you got to realize that when you're the first one there, you don't have to be good. That's kind of a principle of evolution, right? So if you have very sloppy coding, as long as there's nothing else doing coding better than you, you win. And so, you know, now we're in a very sophisticated biological milieu. And so, you know, if something doesn't code properly, it dies. But that doesn't mean, you know, we shouldn't take these things from extant biology and propagate them back to the origin of life. I don't think I've really answered the question, except to say that that it was late. I mean, the ribosome went pretty far before coding. And that there were other things happening before coding. A lot of times people think until you have coding, until you have RNA, until you have a polymerase, you can't do anything. And we really don't believe that to be the case, that there's a kind of a chemical evolution that led to all this. Biology is really just the culmination of a chemical evolution, and that people haven't been interested in studying it because they're so fixated on the RNA world, and the RNA world denies chemical evolution, right? It just says RNA sprung up, which I don't think is possible. I mean, RNA is just this incredible molecule. I could go on for like a week about all the special things that RNA could do. And the idea that it sprung up, you know, through a non-evolutionary process is just, to me, it's not reasonable. Evolution can do amazing things, including create RNA and protein.
Fabrício, está sem som.
Yeah. Could we say that ribosomal protein would suffer an initial selective pressure towards the stabilization of RNA?
Exactly. In fact, we have done these experiments. So what we believe is that the ancestor of protein was what we call depsipeptide, which is a mixture of ester and peptide. And if you make these molecules, they hydrolyze quite quickly. But if you add RNA, the RNA protects them and they protect the RNA. In fact, we think this is the basis of sort of why did polymers start working together? Because they protected each other from chemical assault, from hydrolysis. If you think about biology, it's all about making things through dehydration and breaking them through hydrolysis. I mean, that's everything, all the polymers, everything. That's their story, right? You make them through condensation, dehydration, and you break them with hydrolysis. So we think the roots of chemical evolution were these kind of polymers that were not very good, but they could associate with each other and protect each other for hydrolysis. So therefore, they live longer. In fact, chemical evolution is always about what hydrolyzes the most slowly. So when you make and break things just by hydrolyzing, and you condense and hydrolyze and condense and hydrolyze, because the Earth is turning, the whole surface is getting wet and dry every day. And if some small subset of molecules assembles, their populations is going to go up. And so the initial evolutionary driving force was not templated synthesis, it was protection. Things that live longer had higher populations. And so RNA and protein are the product of these two molecules that came out of this selection for protection. So RNA protects protein and protein protects RNA. They do a beautiful job because that's their roots. That's where they come from. That's our model. I should say, that's our model.
Savio, do you have another point?
Yes, I think that the questions about amino acid RNA synthetases, Fabrício.
So the synthetases, you know, what we can see, I think you asked about the synthetases. If you look in the synthetases, what we see are these same primitive domains. The ribosome started synthesizing these very primitive all beta domains, partly because it protected our ribosomal RNA, but also because they had catalytic function and they were doing things outside. So this proliferation of these primitive beta domain proteins actually, we see them in transcription, we see them in the synthetases, we see them everywhere. They arose, we think, all at the same time. The ribosome figured out how to make probably very primitive and probably not very good beta domains. And, you know, one of the things about folding, if you can make an assembly, catalysis is free. Okay? In fact, it's interesting. There's a guy named Otto who's a combinatorial chemist who makes all these things. And I heard him talk, and he said that same thing. He said, you know, if I can make molecules assemble, catalysis is free. Because assemblies just can do catalysis. It's just, you know, if you can put atoms in three-dimensional space where you want, then catalysis is free. So we think, you know, as soon as biology figured out how to make these very primitive beta domain proteins, I mean, that enabled catalysis, and it went everywhere, and it went to the synthetases. But it's not like these came before. Like everything kind of bubbled up together. We don't think the ribosome came before synthetases, or synthetases came before the ribosome. I mean, all of this stuff was linked. It's just like a lichen where you have an algae and a fungus, and they're co-evolving. I mean, we think these molecules were doing the same thing in these systems. Like, you know, you can't have a ribosome without transcription. You can't have transcription without a ribosome. You can't have any of these things without energy. And so nothing came first. This idea, people in the origin of life are always worried about what came first. I think that's a mistake. Nothing came first. That's not how biology does things. It doesn't have to do things in an order. It can do a lot of things together. So the synthetases and the ribosome, they came together.
Thank you very much, Loren. I think we have a great talk, a good discussion, and participation of the audience here. Thank you
very much for inviting me to this. It's really my honor to be here.
For taking this time to be here with us. No, I mean, we hope to meet you again with a new synthetic model of the evolution of life, maybe. No? Savio?
Loren, thank you very much.
Thank you, Savio. And I like your papers. Savio, I read all your papers. They're great.
Oh, thank you.
Thank you, Loren. So we are now going to go into the closure of the meeting.
Okay.
Thank you for your presence.
I will stop there. Okay. Thank you guys very much.
Thank you very much, Loren. Bye-bye.
Okay. Bye-bye.
Então, boa noite a todos, né? Obrigado aí, todos que participaram até aqui, ou estão vendo aí o vídeo gravado, que vai estar disponível aí no YouTube, no canal do Evolution Day, né? Que também é o mesmo canal do Darwin Day, a gente está chamando tudo canal Origens, né? Então, foi muito bom esse evento, a gente teve uma boa participação, né? Várias discussões, várias ideias aí para os novos eventos, né? Então, a gente está discutindo com a comunidade, inclusive, meios de aumentar, né, a presença de cientistas, principalmente nessa área, né, e de maneira ativa, né, a gente tem que realmente ir contra a maré, né, de alguma forma, com o apoio de vários pesquisadores, né, inclusive na organização. E é isso que a gente vai fazer com o próximo Darwin Day, né, que a gente vai organizar em fevereiro de 2022, tá? Vou fazer só uma apresentaçãozinha aqui. Então, o Darwin Day, né, ele vai ser organizado de 9 a 11 de fevereiro, junto com a Sociedade Brasileira de Genética. A gente já está recebendo sugestões através desses dois e-mails aí, inclusive de palestras de temas atuais, né, com pessoas competentes aí da área, principalmente do Brasil. Né? A gente também deve ter alguns palestrantes internacionais nesse evento, né, mas, por enquanto, a gente está começando a montar, a gente vai ter bastante tempo de organização, né, então a gente espera a contribuição da comunidade para esse Darwin Day, que é sob coordenação da Sociedade Brasileira de Genética, né? Eu estou falando aqui em nome de uma comissão organizadora bem grande, que inclui, inclusive, a diretoria da Sociedade Brasileira de Genética, a professora Ana Benco, a professora Glória Franco, aqui do ICB, da UFMG, tá? E eu queria agradecer mais uma vez aí os meninos que nos ajudaram aí na organização, né, a Alessandra Lima, a Carolina Valente, Flávia Martins, que estão fazendo a disciplina da professora Fernanda Antunes, né, onde também está o Davidson Campos, que também é aluno do nosso laboratório e está nessa disciplina. Agradecer muito aqui a Joaninha, né, aluna de graduação, que está com a gente aí nos Darwin Days desde 2019, né, acompanhando aí essa luta para manter aí uma atividade de divulgação, né, que é gratuita, não tem custos, né, mas é muito difícil de organizar.
A Joana é quase a alma do Darwin Day.
Exatamente. Então, assim, a gente tem tudo que agradecer essas meninas aí que nos ajudaram muito, né, porque foi uma grande correria aí, menos de 3 semanas para ter isso formatado, né, da maneira que foi. Óbvio que a gente cometeu vários erros aí, então eu e o Sávio aqui, né, estamos reconhecendo, né, a importância de assumir esses erros, obviamente, né, e de maneira consciente, né, de uma próxima organização, que vai ser bem próxima, é o Darwin Day, a gente já está abrindo aí uma nova perspectiva, né, para falar um pouco. Então, aqui veio uma colega nossa aí, a professora Glória Franco, tá? A Glória Franco é da Sociedade Brasileira de Genética. Glória, você
Eu
exatamente...
sou tesoureira, segunda tesoureira.
Ah,
Faço parte da diretoria.
ela lida com dinheiro, mas a
Não,
Glória...
quem lida é o primeiro tesoureiro, que é o Reinaldo Brito.
Ah, tá.
Eu
Você
lavo
só...
as minhas mãos.
Não, você só investiga o Reinaldo Brito, né? Mas é que o evento da Day ele vai ser também remoto, né, Glória?
É. Queria aproveitar, então, falar um pouquinho do Darwin Day, se me permitem aqui. Primeiro, eu queria parabenizar o Fabrício, o Sávio e toda essa equipe maravilhosa pelo Evolution Day. Eu acabei de assistir a palestra do Loren Williams, eu estou encantada, absolutamente encantada, foi maravilhoso, e tudo que eu assisti, infelizmente eu não pude assistir na parte da manhã, mas eu fiquei aqui a parte da tarde, eu adorei, e não me pareceu que teve nenhum problema de organização, porque estava tudo muito sensacional. Então, eu queria parabenizar vocês dois e os meninos aí, e dizer também que, por exemplo, o Davidson foi parte da nossa equipe do NAPG, que é o Núcleo de Apoio à Pós-Graduação do ICB, que está aí no Darwin Day organizando também, junto com a Sociedade Brasileira de Genética. E a Joana já se colocou à disposição, Joana maravilhosa, para também ajudar-nos na organização do Darwin Day. Nesse momento, a gente já está com alguns palestrantes confirmados, mas temos ainda muito espaço para outros palestrantes. E o interessante do Darwin Day é que a gente vai ter possibilidade de apresentação de trabalhos pelos estudantes, na forma de pitches. Vocês sabem muito bem como são os pitches, que são pequenos vídeos de 3 minutos ou 5 minutos, para que os alunos apresentem os seus pôsteres. Então, ao invés de ser uma sessão de pôster, é uma sessão de pitch com discussão. A gente fez assim no último Congresso Brasileiro de Genética. Nós também temos os Drops of Science. Esses dois são modelos que a gente usou na mostra do ICB, que foi realizada aí recentemente pelo ICB, com apoio dos vários núcleos do ICB, dentre os quais o NAPG, e os Drops of Science serão palestras um pouco maiores de pessoas interessadas em fazer comunicação do seu trabalho. E essa comunicação pode ser relacionada à pesquisa, à educação, né? Sempre com o pano de fundo da evolução. Então, vai ser muito interessante. Normalmente, no Drops of Science que a gente fez na mostra do ICB, as pessoas que mandaram suas palestras eram alunos de pós-graduação, doutorado e pós-doutorandos. Mas a gente está abrindo no Darwin Day para todas as modalidades, são apresentações de 10 minutos, e você terá a condição de apresentar melhor o seu trabalho ao invés de um pitch. E as melhores palestras serão selecionadas para serem apresentadas no Darwin Day. Então, além dos palestrantes convidados, nós teremos esse espaço aberto a todos, estudantes de graduação e pós-graduação, para mandarem os seus trabalhos.
A ideia dos Drops of Science foi uma explosão de criatividade, né, dos alunos aí. Eu imagino que isso...
Já vem há mais tempo, né? Começou na mostra do ICB anteriormente, mas os drops, eles eram para professores, e aí a gente mudou um pouco o foco, colocando ele nos estudantes. Por exemplo, o Tomás Pinotti foi um que falou na mostra do ICB, do Drops of Science, foi um dos 6 selecionados para falar na mostra do ICB, sobre os chicletes de Neandertal, que foi maravilhoso.
Que bom. Então, a gente espera, com essa parceria aí, várias instituições, né? Tem a SBG encabeçando, mas tem o NAPG, né? Tem a gente aqui como professores, o Sávio, eu, né? A Glória, toda a diretoria da SBG. Estou faltando, quem mais?
Ana Benco, né? Que é a presidente da SBG.
Ana Benco, sim, toda a diretoria, mas
diretoria.
E tentar fazer um Darwin Day.
E a equipe dos alunos do NAPG, que são os que nos ajudam na organização dos eventos. O Davidson foi um membro, mas acabou saindo recentemente por causa das suas atividades na pós-graduação, mas nós temos uma equipe extensa de alunos de vários programas de pós-graduação do ICB. Quem for interessado em contribuir nessa parte de eventos, a nossa equipe está disponível, temos muito interesse em termos alunos criativos, que tragam sempre novidades, que gostam dessa parte de divulgação e organização de eventos. Vou até entrar em contato com a professora Fernanda para ver o que a gente poderia fazer junto e ter novas ideias, né, para a divulgação dos nossos trabalhos no ICB, das pós-graduações do ICB.
Uma reivindicação que a gente tem escutado bastante esses dias, né, na verdade já tem um tempo, mas é ter uma participação maior da sociedade acadêmica, até na elaboração do programa, né? Eu acho que seria uma boa a gente abrir algum canal no Instagram da SBG, algum outro lugar, além dos e-mails que tem, né? Justamente para novas sugestões de temas que às vezes escapam da gente, das nossas conversas, né? Que a gente fica nessa ideias herméticas, né? E a parte de ensino também, né? Que a gente aí espera uma participação maior dos professores e estudantes de ensino médio também, em algumas atividades.
Com certeza. Eu acho uma ótima ideia, Fabrício. Sempre sugestões da comunidade são muito bem-vindas. Eu posso levar essa reivindicação à diretoria, né, e de criar um espaço no Instagram, mas, a princípio, nós estamos recebendo sugestões pelos e-mails. Então, está aí o contato da SBG ou o e-mail do Darwin Day, darwinday.uf.fmg@gmail.com. Podem enviar para a gente, serão muito bem-vindos essas sugestões.
Pois é, então estamos chegando ao fim aí. Obrigado, Glória.
Obrigada a vocês pelo espaço.
Pelo apoio, participação também aí nas perguntas, né? Essa palestra do Loren, né? Ele trabalha com uma complementação do que a Ada Ioná, que foi a palestrante do Darwin Day, que era o prêmio Nobel, né? Que ela trabalhou com evolução de ribossomos, e ele faz algo complementar. E foi bem interessante hoje, porque ele mostrou um sonho antigo, a questão da síntese, né? Para simular evolução química no início da vida, que era coisa muito complicada, né? O pessoal mal estava conseguindo síntese de RNA, né, Sário? E agora ele está conseguindo coisas mais complexas ainda, né? Eu estou curioso para ver essas
Maravilhoso. Eu tenho até uma sugestão, a gente podia convidá-lo para o próximo evento da Sociedade Brasileira de Genética, que será presencial ou talvez híbrido.
Esse é o de agosto.
Em setembro. Setembro. Setembro.
Glória,
início de setembro.
Que ele vai aceitar com muito prazer.
Nossa, eu adorei. O Sávio vai ser, assim, um estouro. Eu achei, assim, sensacional, muito inspirador, muito.
A gente esteve com ele aqui em 2014, Fabrício?
Foi em João Pessoa, né?
Foi,
em João Pessoa,
2014,
quando a gente organizava os eventos aí presenciais, né? Então, o Loren, a Ada e a Ioná, né? Todos participando, e assim, no pessoalmente, com essas pessoas também é outra coisa, eles são super amáveis, eles gostam de realmente participar das conversas, né? Então era muito interessante esse tipo de ambiente, que está em falta, né? Está todo mundo sentindo falta dessas coisas.
Né? Então, eu queria pedir a ajuda de vocês para a gente formalizar o convite a ele.
Perfeito. Perfeito, mas vamos pensar em vários outros nomes aí. Tá bom?
Claro. Nós temos várias áreas temáticas no Congresso, né? Evolução é uma das áreas temáticas. Bioinformática, eu acho que a gente está precisando de mais palestras na área de bioinformática. A gente tem muito, assim, genética molecular, genética humana, evolução tem um pouquinho, mas talvez a gente estamos em conversação para fazermos juntos com um evento de bioinformática mesmo, da Sociedade, da Associação Brasileira de Bioinformática e Biologia Computacional. Então, assim, se der tudo certo, a gente vai fazer o evento junto, vai ser em Campos do Jordão, então vai ser ótima ideia de a gente trazer o James, o Loren, desculpa.
Vai dar certo, sim. Então, muito obrigado. Alguma palavra final, gente?
Fabrício, eu quero só fazer uma propaganda aqui, deixa eu fazer rapidinho. Prometo não tirar muito seu tempo. Que é de um evento que vai ter. Você consegue ver minha tela?
Deixa eu ver.
Tá
Sim.
vendo, Fabrício? Tá.
Sim.
Essa é uma rede que eu faço parte lá da Inglaterra, que é uma rede de pesquisa em evolução química. E a gente está expandindo os grupos de ação, né? E vai ter esse evento aí, dia 8 de janeiro, com várias palestras aí interessantíssimas com problemas, como a ciência pode tentar resolver alguns problemas que a gente tem no planeta hoje. Vai ter gente da física, da astrofísica, da astrobiologia, que vão discutir várias temáticas. Aqui está o link, é totalmente gratuito, não precisa pagar nada. E então, quem tiver interesse aqui, ó, eu sugiro, porque realmente os eventos pelo Nocel têm altíssima qualidade e sempre trazem um pessoal muito interessante das mais diversas partes do planeta, para ver visões totalmente diferentes das coisas. E uma última coisa, Fabrício, que a gente esqueceu, a gente tem 2 livros para sortear. Eu não sei como a gente faz isso, pelo número de inscrição.
Vocês têm, eu tenho uma sugestão. Vocês têm lista de presença?
Temos.
Sim. Usa um sorteador da internet com um número da lista de presença. E aí você pode sortear um livro para a lista de presença da manhã, um livro para a lista de presença da tarde. Foi assim que a gente fez na mostra do ICB.
Perfeito, a gente vai fazer assim, mas a gente vai mandar por e-mail depois aí.
Pronto, a Joana, vou me encarregar a Joana de fazer uma live do sorteio.
Ah, é? Exato.
Isso é legal, Uhum.
A gente faz isso. Então tá, gente. Muito obrigado por tudo, todo mundo que participou, todo mundo que vai ver aí a gravação, né? A gente espera aí ter contribuído um pouco para aumentar o conhecimento, o interesse pela ciência, né? Que foi isso que nos motivou a fazer biologia, né, Glória? Depois pós-graduação, passando vários maus bocados, né? Vários problemas como todos, e agora a gente está passando um que a gente nunca imaginava ter passado. Nunca imaginado que passaria, né? Se voltasse aí 10 anos, quem ia falar que aconteceu algo assim, né? Mas a gente vai continuar aí de pé firme, né? E trazendo mais ciência para a sociedade, né? E vamos tentar melhorar cada vez mais, né? E óbvio que sempre tem muita coisa para melhorar, e agora a gente está ciente de vários problemas, e com isso, né, a gente conscientemente vai tentar melhorar um pouco também a questão da igualdade de gênero, né? Que a gente conversou hoje em alguns momentos aqui, né? E foi, inclusive, reivindicação do Mulheres na Ciência, né, Glória? Você que também participou disso, né? Então, a gente espera melhorar nesse assunto também. Bem, é isso aí. Então, tchau, gente.
Tchau, gente. Prazer. Fiquem bem.